Hey Team Bio, if you're in 5-6 section, I added a few images to this document. So if you want to come by my office and pick up a new copy of the document, feel free. There are only 3 additional images, but it might be useful to you to add these to the doc that I gave you in class today. Okay, so we're going to be talking today about protein structure, and we're going to be talking about the relationship between structure and function. That's going to be the theme of today's screencast. Today in class we performed a bunch of dehydration synthesis reactions. You know the common backbone of an amino acid. We have an amino terminus, which is a nitrogen bonded to 2 hydrogens, and then we have a central carbon. The central carbon is also bound to a carboxyl group, which looks like this, and a hydrogen, and then a variable group. Okay, you're all going to hate me. I apologize. I'm going to copy this, paste it again here, and I'm going to take this OH group and turn it blue, and this hydrogen style, and also turn it blue. You know that if you bring these together to form a dehydration synthesis reaction, synthesis, what happens is you pull a water out of this, copy, paste, grab this, boop. And just for ease of, I need to make this eraser smaller, of drawing, I'm going to draw it like this. And then H here. What happens is we get a water and a dipeptide. So this is our dipeptide, and water, and this bond between the amino terminus of this amino acid with the carboxyl end of this amino acid, this is called a peptide bond. Okay, so if we do this reaction many times over, we create a polypeptide. So this is my best interpretation of a polypeptide. This is a polypeptide. And I've kind of drawn it as 2 words, but know that it's 1 word, polypeptide. Each of these different colored dots represents a different amino acid. So maybe this is methionine, and this is phenylalanine. I think it's PHY. I'm not sure. Anyway, they all, or PHE is phenylalanine, or alanine, or valine, so on and so forth. They're all joined together by these peptide bonds. And this peptide bond is a covalent bond. So it is very strong. The sequence of amino acids, the fact that this is methionine, followed by phenylalanine, followed by alanine, followed by valine, so on and so forth, is called the primary structure. So this is the sequence of amino acids. And it's joined together, as I said before, by peptide bonds, and these are covalent bonds. They're very strong. Okay, but you know that this linear structure, this primary structure, is only a polypeptide. It's not yet a protein, because in order to get a functional protein, we need to take on a 3-dimensional shape, which can only be formed through folding. And that's where secondary structure begins. So there are a bunch of different secondary structures, but what I'm going to focus on is the 2 structures called alpha helices and beta sheets. So the two images that I added to this drawing, or two of the four, are this and this. I was just thinking that maybe this drawing wasn't quite enough. Okay, what I was trying to do with this drawing here is show you that if you take a linear chain, so if you take your primary structure and then you fold it into a alpha helix, into like a spiral staircase pattern, you'll get an alpha helix. If you fold it into alpha helix, you get alpha helix. And the way the secondary structure is maintained is through hydrogen bonding between the amino acids within the helix. So we will get, for example, maybe a hydrogen bond between these two, and 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 these two. So, so you can see here we have the carboxyl, or what was part of the carboxyl group of one amino acid is binding to an amino group in another amino acid further down the loop. So alpha helices are held in place by hydrogen bonds. And likewise, beta-pleated sheets do the exact same thing. So if we took a linear chain, a polypeptide, and we folded it into this sheet-like pattern, like so, we would get what's called a beta sheet. And just like the alpha helix, these beta sheets are held in place through hydrogen bonds across the sheet. And these hydrogen bonds will be at this throughout this whole thing. So you can see here are the hydrogen bonds between the amino terminus of one amino acid with a carboxyl group of another amino acid, or what was once a carboxyl group. So these beta sheets are also held in place by hydrogen bonds. So when we start to take our polypeptide, our primary structure, and fold it up into a more complicated structure, into secondary structures, alpha helices or beta-pleated sheets, these forms are maintained by hydrogen bonding. Now, remember, hydrogen bonding is a much weaker bond than a covalent bond. So these can be much more easily broken. That's going to be important to keep in mind. Okay, now we move on to tertiary structure. So proteins, when they are functional, are not only composed of alpha helices and beta-pleated sheets, but also they're stabilized by other interactions. So tertiary structures are when you take multiple secondary structures and they interact with one another. So what I did here is I took my alpha helix and I added it to my beta-pleated sheet. But a more accurate representation of what's going on is something that looks more like this. So you'll see this structure has a beta sheet right here. So this is a beta sheet. But this is also a beta sheet here. And this is an alpha helix. And then in between these beta sheets and alpha helices are other folds of the amino acid. So what this picture is lacking, is this idea that all of these squiggles are actually different amino acids joined together by peptide bonds. But the more complicated folding structure is held together by these bonds. So the bonds that hold together tertiary structures are hydrogen bonds, So they contain alpha helices and beta-plated sheets. So those, as you know, are held together by hydrogen bonds. And so tertiary structure is in part going to be held by hydrogen bonds, but it's also going to be held together by clustering of hydrophobic R groups, often in the center of the protein. And the reason for this, the reason why the hydrophobic R groups are often found at the center of the protein is that all of these proteins are living in aqueous solution inside cells. That means they're living in a watery solution. So the hydrophobic R groups tend to end up in the center of the protein because it's a lower energy state for the water-hating side groups, the R groups, to be away from the water, whereas the outsides of the proteins are often studded with hydrophilic or charged or polar R groups. So, charged or polar R groups around the outside. And the last form of structure that, or the last type of bonding that holds together tertiary structure is called disulfide bridges. Disulfide bridges are bonds between two sulfur atoms. Oops. And this is a covalent bond. So it's pretty strong. But tertiary structure, much like secondary structure, is mostly held together through weaker interactions. So this hydrogen bonding, this clustering of hydrophobic R groups in the interior, and clustering of polar R groups around the outside, these are all pretty weak bonds. So tertiary structure, much like secondary structure, is a lot weaker than primary structure, and it can fall apart. Okay, quaternary structure. Quaternary structure is just like tertiary structure, except that it involves multiple subunits. So here I have an example of, this would be a tertiary structure protein. It's composed of one long chain of amino acids. So if we were to unravel this, these would all still be connected together. What differentiates quaternary structure from tertiary structure is that quaternary structure is composed of multiple subunits. So here is one subunit. And here is another subunit. So some proteins, their highest level of structure is tertiary structure. They're only composed of one subunit. But there are many proteins that, in order for them to be activated, require the association of multiple subunits. So you can see here, there are 4 subunits, 1, 2, 3, 4, in this final protein, just like this protein has 1, 2, 3, 4 subunits as well. So just like tertiary structure, the quaternary structure is often held together by hydrogen bonding between the subunits. Okay, so that is quaternary structure. The big takeaway from this screencast is the idea that secondary through tertiary structure and all the way to quaternary structure, so I guess to say secondary through quaternary structure, is all held together by relatively weak bonds. And we're going to talk about the different ways if you disrupt hydrogen bonding. The protein structure. Will fall apart. And the protein, which takes on this three-dimensional shape, will return to the primary linear structure. And when it's in this form, it can't do its job of catalyzing reactions. So keep that in the back of your head. Proteins need to take on a three-dimensional shape, which is mostly the three-dimensional shape is mostly held in place by hydrogen bonding. So if we do anything to disrupt hydrogen bonding, that protein structure will fall apart and it will cease to function as it should. Okay, that is all.